Hey guys, this is an important topic, the mitochondria and chloroplast. Both are really important. We're going to spend an entire chapter on the mitochondria and an entire chapter on the chloroplast. So a lot of information coming on these two important organelles. If you look at what the mitochondria is, the mitochondria and chloroplast are essentially ancient bacteria. Why we say this is that they have not just a cell membrane, but a cell wall like bacteria do. They have circular DNA. That circular DNA thing, only bacteria have circular DNA and these two organelles. And they have ribosomes, and the ribosomes have the molecular weight that you only find in bacteria. All of this is just further evidence that the mitochondria and the chloroplast were once free-living bacteria a long time ago. So let's look into this. So the mitochondria, its main purpose, other than being the powerhouse of the cell, is to take glucose, C6H12O6, and in the presence of oxygen, it will break it down to make H2O, CO2, and of course, the ATP from that reaction. That ATP from the reaction is the very thing needed to drive all of our cellular reactions. So we live by this mitochondria. The mitochondria, that is, there's many of them in every one of our eukaryotic cells. And so that is huge in what its purpose is. It's providing energy so we can have those chemical reactions going on, so we can maintain our entire being of chemical reactions. The chloroplast. The chloroplast itself is going to be the same thing with the bacterial background, but it's going to be photosynthetic. We probably knew that, but it's going to take water, and it's going to take carbon dioxide, and it's going to produce a carbohydrate, C6H12O6, as well as O2. So the process of cellular respiration versus the process of photosynthesis, they're essentially reverse of one another, is the way you can look at it. We'll look at it in more detail later on. So these are two very important organelles. So the mitochondria and chloroplast, they display similarities that are going to be similar to that of a bacteria. So they essentially, I want you to think of them as bacteria that are trapped in eukaryotic cells, that they live their life in eukaryotic cells, and that benefits the eukaryotic cell by them existing in it. This process of living within, like inside of, it's called the endosymbiotic theory. And I do expect you to know that. The endosymbiotic theory points to that the certain bacteria, like the mitochondria and possibly the chloroplast, depending on the cell type, is going to be consumed, held into the original cells that started with this. And to give you an idea of how beneficial this is, there are really no cells on Earth, essentially, that lack the mitochondria. So all cells are essentially going to have the mitochondria inside of them. Eukaryotic cells that were without a mitochondria were outcompeted by the existing cells that had random mitochondria. And therefore, now the winners are all the mitochondrial cells. So that is the endosymbiotic, also called the endosymbiote theory. Endo is referring to inside of. Symbio is referring to same life theory. So this is the theory that something is inside of that cell, and as a result, that cell can survive. So if you read through these bullet points and kind of get an overview, and if you need to pause me, that's fine, but I definitely need you to make sure that you're getting this, and that is that you had ancient bacteria that were consumed by an ancient eukaryotic cell, and that eukaryotic cell that had that mitochondria inside of it had a much better chance of living, because the byproducts of that mitochondria were ATP. It just existing was releasing the very energy need that cell needed. So it was a major benefit for something to randomly have this. And if that thing had that and that energy was being well provided, those cells that were dying just randomly weren't dying off as much because they had that mitochondria inside. So since the mitochondria can reproduce like bacteria by binary fission, they can make more of themselves. And when the cell becomes two, now there are mitochondria that are now in both of these. And so they're going to just continue to pass on more mitochondria to each generation that is going to come from each one of those cells. A few things that we can notice with the mitochondria. You can see these lines back and forth. Those lines back and forth are referring to something called the cristae. So the cristae, I'll try to get you to the point of view. The cristae are going to be these curvatures, and these curvatures are very important. Remember that I talked about the accordion, that you have that curvature that goes in and out and in. So something that is this distance really measures to be this distance. And that is because the curvature that's there is essentially squashed down to a smaller area, but due to all the curves, it actually measures as if it is this large. And so since it is that large, you can see that it's going to have a lot of surface area. So if you look at the cristae, it's going to be a big part of forming the actual matrix of that mitochondria. And so take a look at this. You can see all those different curvatures, and here is the transmission electron microscope image. You can see all those different lines of the cristae, and you can see all those inner folds that we have there of the cristae of this mitochondria. And you can see that we have the outer layer peeled off so we can see that inner membrane. And between there is the inner membranous space, and you can see the inner membrane versus the outer membrane as they are identified. But key thing is here, surface area. Surface area, this is allowing this organism to work as if it was in a much bigger cell. All the extra surface area essentially makes that bigger. So chloroplast. A chloroplast is going to be, we know that they're green because of chlorophyll, but a few things that we have in them is we're going to have that light pigment, that chlorophyll that can receive sunlight as it moves down and hits that cell. And since it hits that cell, it's going to go through the process of photosynthesis. A few of the components that we have in this are the thylakoids, the granum, the stroma, and we're going to identify those in the next piece. And so if we look at the thylakoids are going to be essentially a small region of this, as you can see right here, that small little disc, and it's even bracketed out to be shown right there. And we can see that little bracketed region, which is important. With this whole stack here, that whole stack is going to be called the granum. Initially, when scientists were looking at that, and you can kind of see over here, they thought that these were grains. And so when they were naming it, they named it essentially grains. Later, they found out that they actually weren't grains, so they stuck with it and we're just going to keep that name. So that's granum. So granum is going to be the stack of thylakoids. And if you look further, you can see something else that's here as well. See the circular DNA? The only place on Earth you have circular DNA is a prokaryotic cell. Here we have it in an organelle. That's the only organelle that we have that has DNA. All the other organelles, other than mitochondria, so those two are the only two that have DNA, and it's circular DNA, which is further evidence. So if this is in a court of law and you're trying to really determine that these are from this area, you would need evidence, and that's what we have. We have circular DNA. We have the double membrane, including the cell wall. We have the ribosomes that are of that mass. They're going to reproduce their binary fission. All of those characteristics are things that you only find in bacteria, except for right here, which is where you find it in the chloroplast and mitochondria. And that is further evidence that the endosymbiotic theory is supported. So something that I hadn't mentioned yet, and that is the stroma. The stroma you can think of as the cytosol of this. It's going to be the liquid matrix that everything is going to be suspended in, and that is the stroma. See transmission electron microscope, TEM, that's showing through those different pieces. Here we have the different chloroplasts that are shown in red in an algal cell. Something else we want to look at is something called peroxisomes. And peroxisomes themselves are going to be a specialized single membrane, and they're going to produce hydrogen peroxide. So H2O2 is that hydrogen peroxide, and as a result of that chemical reaction, it's going to release water as a byproduct. And this is going to be used like lysosomes to break down certain things by using that chemical reaction to damage that tissue. All right, guys, so that sums up 4.5. And make sure you are very familiar with the endosymbiotic theory. It is important, and it shows up several different times throughout the year. All right, stay safe.